Okay, so this is going over the problem set, the PowerPoint slides for the exam to review. I'm just going to leave it in this format so you guys can see the question, and then the answer is A. And so let's figure out why the answer is A. So we have our parental gametes. I'm just going to write on this slide. So we have the parental gametes, which are big A. Big A. Little b. Little b. And that's crossed to little a. Little a. And big B. Big B. So when we have this cross, the gametes that this can produce has to be big A, little b. And the gametes this can produce is little a, big B. Right? And they have to be in this order of the chromosome. There's no other way to do it. Even if we have a crossover event, it would be the exact same thing. So anyway, this is the makeup of the chromosome. And that's what we show here. So the gametes from this we could produce would be, from this offspring, would be big A, little b, little a, big B, which would be the parental ones, right? So the parental ones would be big A, little b, little a, big B, right? And then there's really no other combination of that. The question asks which pair of gamete genotypes are recombinant, and the answer is that if this is the genetic makeup of the parentals, which is right here, and this one is the other parental, which is here, then recombination would create big A, big B. Whoops. Sit over here. Big A, big B, one chromosome, and then we would get this chromosome and this one, which would be little a, little b. So this matches this, and therefore the answer is A. Make sense? Hopefully. All right, let's go to the next one. So the next question is, the following testcross is made between two cord plants, big R, little r, big S, little s, and then this is the tester because we know it's genotype. So what's the distance between these genes? Well, this is the parentals because it's the largest number, and this is the recombinants because it's the smallest number. So in order to calculate this, all we have to do is add up the recombinants and divide that by the total number. And that gives us 100, and this is 100. I'm guessing that that's going to be 900 since that's how I usually set these up. Yeah. So that's 100 divided by 1,000, which is 0.1, which is 10%. And remember, the percent we just convert to centimorgans. So the answer is 10 centimorgans, which is C. All right. And then that just shows how we just did that. All right, we're going to do another test cross. Different pair of corn plants. We want to know the distance between the genes S and T. Again, we do the same thing. So we take 14 plus 16, divide that by 14 plus 16 plus 487 plus 483. And that gives us 30 divided by, let's see what this is, 487 plus 483 plus 30 is 1,000. So that is 0.03, which is 3%, which would give us 3 centimorgans. So the answer is B. And that just shows you how to do it. All right. So this question is the distance between R and S is 10 centimorgans, right? So we have that right here. Distance between S and T is 3 centimorgans. Based on this data, which map is correct? Well, the answer is that you don't know because we haven't tested R and T, which we have to do to figure out if that distance is 13 or if that distance is 7. So the answer is C. Is not enough information to be able to tell. Okay, next. This is just showing you the way that you could figure this out is figure out the distance between R and T. Once you have that, then you would know which one of these maps is correct. 2 genes A and B are 8 centimorgans apart. If you perform a 2-point test cross for A and B, what percentage of the offspring do you expect to be recombinants? Okay, so this is the offspring, and that means that that's going to form a zygote. You have to pay attention to the question because it may say gametes, and that's different. So we're going to fully do this out. We know that we have we're doing a 2-point test cross for big A and big B, and then so 2-point test cross is a heterozygote to a homozygous recessive. So the gametes that would be the parental ones we would expect to be these, and the recombinants we would expect to be big A, little b, or big A, little b, or little a, big B. All right, so, but it doesn't even really matter because what we know is that 8% of the recombinants offspring are going to be the progeny because 92% would be the parentals, not non-recombinant. So the answer is 8% B. You don't even need to do this. Next. C and D are 86 centimorgans apart. That's not, you can't tell that, right? The rule is 50, if it's 50% or greater, then it looks like independent assortment. So the answer would be D. You can't tell because that's the highest value that you can get. All right, next one. Herbicide resistance is recessive trait, which is lowercase h, so that's recessive. Wild type means that if you put an herbicide like Roundup on you, then you die. This means you resist Roundup. Purple flower color is recessive. It's kind of weird, but okay. And the dominant wild type allele produces yellow flowers. So yellow is dominant to purple. I did that on purpose just to make you think about the question so you didn't just blindly go, oh yeah, purple is dominant. So H and P genes are linked at a distance of 18 centimorgans, a plant with genotype. All right, so let's just write the genes down. The plant is H plus and the other one is P plus. We know they're together on the same chromosome because that's what this line shows us. And then the other one is little h and little p. We test cross that. We know that when we do a test cross, it's to the homozygous recessive for both traits. All right, so what proportion of the offspring would you expect to have yellow flowers and be susceptible to herbicides? So to be susceptible, they would have to have at least H plus. All right, that's susceptible to herbicides. And they would have to have yellow flowers, they would have to have at least a P plus allele. All right. So the gametes that are going to be made, I'm sorry, should be a little p. All right. So the gametes that are going to be made by the test crossed are always going to be the same. Even if there's crossing over, it's still little h, little p. The gametes made by this, the parental gametes, the ones that aren't due to crossing over, are going to be H plus and P plus, or so this is one, the non-recombinant genotype. The other one is here, and that would be HP, HP. So these flowers here, erase this. Okay. So these plants here would have H plus and P plus. That means that they would be yellow and herbicide susceptible. All right. So it's 18 centimorgans apart. So we would expect these gametes would be produced at a rate of 1 minus 0.18, which is 0.82. So 82% should be this. And then we can divide that by 2, which would give us 41% for this trait. All right. So now we're going to go back. And we're going to do the other ones. So we know that this gene is linked. It's H plus. So the gamete has to be, the chromosome is H plus, and P plus. This is the parental, H and P. And so if we have a crossover event, then we would end up with this gamete, which would be, running out of room here, H plus P, right? And then that would be the other parent is the 2-point test cross, so the only thing it could give would be the recessive genotype, and then the other recombinant would be P plus, right? And then HP. So this would be this one would be herbicide susceptible, but it wouldn't have yellow flowers. It would have purple flowers. This would have yellow flowers, but it would be resistant. So the question asked, what percent do we expect to have yellow flowers and be susceptible to herbicides? And the only one is this one right here, which would occur at a frequency of 1/2 of the 82%, or C, which is 41%. So that's the answer. All right, on to the next. This is asking, what portion of the progeny would you expect to have the recessive phenotype for both traits? So if we set up a standard Punnett square here, I'm not going to fill in all this stuff. You know the only way to get a progeny that is recessive for both traits is making these gametes, which is little r, little s. I know these are terrible letters to pick, but this is little r, little s. And then the end we would get little r, little s, little s. And if these are sorted independently, we would expect this to be made 1/4 of the time, and this to also be made 1/4 of the time. But they're linked. They're not independent. So we can't rely on this number here. We have to calculate this value. So we're going to get rid of that. Oops. If they were both 1/4, if it was independent, then we would calculate the probability of making this zygote, this fertilized egg, as 1/4 times 1/4, which would be 1/16. And the decimal percentage of 1/16 would be 1 divided by 6. 1 divided by 16 equals, that would be 0.0625. All right. But that's not right, because they're not going to be made at 1/4% because they're linked genes. They're not independent of one another. So now we have to figure out what the probability is to make this gamete and what the probability is to make this gamete. And we can do that by what the question says. It says it's 20 centimorgans apart. So we know that if we draw out the parent's genotype, big R, little s, little r, big S, we're crossing that to big R, little s. This is little r, big S. So the parents are the same genotype. Now, what's the probability that this would make the big R, shoot? That's a little s. Well, this tells you 80% are going to be parental and 20% are going to be recombinant. So what's the probability of making this parental one, which is here, is 1/2 of 80%, or 40%. Make sense? And same for this parent, right? This would be their parental, and we could do this across on each square. This side for mom, this side for mom, this side for dad. But we can't get little r and little s this way from parentals. The only way we can get little r and little s is from a crossover event. So what's the frequency of crossover? What are the progeny? 20% are recombinant. So what's the probability we would make this gamete? Well, if making both of these together is 20%, then the probability of making little r, little s is 1/2 of 0.2, which is 0.1. So this gamete is going to be made only 10% of the time. And this gamete will be made 10% of the time. And so just like we did to get 1/16 here, because we had a quarter and a quarter, which would be 0.25 or 25%, and this one would be 25%, but they're not, because they're linked, they're only 10. So to find this out, we multiply 0.1 times 0.1, which gives us 0.01, and that is the answer of making this zygote, which is 1%, which is D. All right, next question. Oh boy. Okay, so this is a chi-square. You guys didn't know how to do this before, so basically we need to know, we have our observed. This is our observed. We need to calculate our expected. If the genes are linked, then we would expect a deviation from the 1:1:1:1 ratio. So each of these should be 25%. So to do this chi-square, we're going to just see if the genes are not linked, and if they're or if they're linked. And the easiest way to do that is to calculate the expected ratios at 25%. So we have to add all these up. I hope if I did this right, it should come out to 100, but it looks like it's not. It's like a 210 or something weird. 56 plus 43 plus 54 plus 47 equals 200. No, it is 200. Sweet. So if that's true, then we would expect our ratios of each of these to be 50. Right? We have 200 offspring times 0.25 gives us 50. So that's our expected. This is our observed. And now we just plug in the numbers for the chi-square. So we have to do each one of these individually and add them up together. So observed minus expected squared over the expected, which would be 56 minus 50, which is 4 squared, which is 16, divided by our expected, which is 50. So 16 divided by 50 is 0.32, and so we'll do the next one. I'm just going to erase this because we don't need it. So we've done short ebony. Then we got 0.32, 0.32. All right, so the next, log gray. So now we're going to do short ebony. That observed is 43 minus 50, which is 7 squared, which is 49, divided by 50. And that's 0.98. All right, so we have, this is long gray, this is short ebony. I'm going to erase this, and then we'll do the next one. So we have 54 is our observed minus our expected, which is 50, which is 4 squared, which is 16, divided by our expected, which is 50. And that is the same value as this. So I'm just going to cheat and call that 0.32. That's for long ebony. And then the short gray ones would be, wait, no, it's not. Let me calculate it out. Dang it. Okay. So 16 divided by 50 equals, wait, did I do that right? 6. I had 56 minus, that's 6, which is 36 divided by 50, which is, all right, this is wrong. This should be 0.72, sorry, because that's 56. And 6 squared is 36, and 36 divided by 50 is 0.72. The other one is 0.34, because that difference is 4 squared, which is 16 divided by 50, which, oh, dang it. Sorry, it's 0.32. And that is the long ebony. And then the last one, the difference between those two is 3. 3 squared is 9, so 9 divided by 50 equals 0.18. So there's 4 outcomes. Our degrees of freedom are 3, right? 4 minus 1 equals 3. This is our critical value. We're looking for a number that's less than that tells us that they're not linked. Any number that's greater than that tells us they are linked. So we add them together, 0.18 times, or 0.18 plus 0.32 plus 0.98 plus 0.72, and I can already tell you that that value is 2.2. That number is smaller than this number. So does the data show that genes are linked? The answer is no way. That is not, those are not linked genes. Those are independently assorting genes. Or they're genes that are so far apart on the chromosome that they appear like they're not linked. That means they're 50 centimorgans or greater. All right, on to the next. Okay, so we have the parents. They're, just on this side, I'll put this parent is big C, big A. We know that because of the nomenclature, the way that it's written with the line there. And then this, dang it, so I'll make this a big C, little c on the next chromosome, and little a. All right. This one is big C, big A, little c, little a. All right. So if these parents, they have all these kids, right? And we're looking for the markers for big A and little a, right? So we can do the non-recombinance, right? And remember, little c means that if you're homozygous for little c, you have cystic fibrosis. So you have to be little c, those are small c's, equals the disease. All right. So if this is a non-recombined gamete, so is this. This is a non-recombined gamete, and so is this. So if this sperm fertilized this egg, we would get big C, little c, and big A, little a. So this would be little a, and not have cystic fibrosis. Where are we at? Oh, sorry. Big A, little a, and not have cystic fibrosis. So big A, little a, right there for the markers. No cystic fibrosis because it shows it there. All right. So that would be this offspring, that cross. This fertilizes this egg. I'm going to erase this here. So then if this fertilized that egg, we would get big C, and big A, big A. Did we see that? This fertilizes this one. So this person would not have cystic fibrosis, and it would be homozygous dominant, which would be this right here. Remember, we can't tell the difference because the gel's only determined by size. So if they're identical, then we only get one line. And this individual would have that. In fact, these are also big A, and big A, right? So which child shows evidence of recombination? All right, so if this fertilized this, right, then we would have, and I'll erase this. So this sperm fertilized this egg here. Then we would get little c, little c, little a, little a. This child would have cystic fibrosis. So none of these children have cystic fibrosis, but this child here has little a, which is weird because that's the only way that you could get little a, because if this fertilized this, then you'd get big C, big A, little a. So this child right here is unusual because this child, if it has little a, it should also have cystic fibrosis. The only way it can get this is if this crosses over and we make this gamete and this gamete, right? So then that child becomes heterozygous for cystic fibrosis, but homozygous for the little a allele. So the answer here is 2, 3. Which is this child right here, number 3, which is C. All right, next. Polydactyly. So we know that has variable penetrance and variable expressivity. It's autosomal dominant. And then we talked about this in Extra Fingers and Hose. Red symbols show people with the trait, which person shows evidence of incomplete penetrance. Okay. So it's dominant. That means that this person we'll just use P. So this person is, well, I'll just use the normal nomenclature. So let's say this big P, right, means you do not have the polydactyly gene. Big P, this little p is the polydactyly gene. But you only need one copy, and then you get polydactyly. So this is saying that red symbols show people with polydactyly. So this person has polydactyly right here. This person is normal, does not. Okay, so when they have offspring, we have big P, and big P, little p. So we would expect 1/2 of their offspring would be heterozygous, but they would show the polydactyly trait because they just need one of the alleles, which is what we're showing here. They had 2 kids. One of those kids have polydactyly. I'm sorry, they have 1 kid. This is an out person. So remember, we assume that this person, we know this person is big P because they don't have polydactyly, but we would assume that anyway. Let me erase this. All right. So this cross we're looking at here, this person is big P, right? They can't be little p because this parent would have polydactyly too. So we can set up our Punnett square again. We have big P, which is this parent here. And then this one would be big P. So we would end up with big P, big P, big P, big P, big P, little p, big P, little p, right? So this person here is unusual. We would expect that everyone in this family to, if it was complete penetrance, anyone that got this would have polydactyly, and if you didn't have polydactyly, you wouldn't have the allele for polydactyly. So in this case of these parents, they should be big P, big P, and that means they couldn't have a kid that's big P, little p right here. Does that make sense? So this is weird. this means that in one of these parents, this parent right here, we would assume that that parent has to be big P, little p, but it doesn't show the trait. It doesn't have that parent doesn't have an extra digit. So this is a carrier of polydactyly, but it doesn't show any penetrance. So it's incomplete penetrance in that individual there, which is, I don't know where we get, oh, there's B. So 3, 2, 3. Let me plug this thing in. Sweet. All right, we're plugged in. So anyway, this parent right here would have to be heterozygous. So we would suspect that this person shows evidence of incomplete penetrance. All right, next one. And that's just showing you that this child couldn't be born big P, little p, unless one of these parents had little p in it. So that's not possible unless this is due to incomplete penetrance. Okay, and petunias, purple color is dominant over white, so classic. Penetrance of the purple phenotype is 50%. So 50% penetrance. That means 1/2 of the offspring that inherit little p, 50% will be white and 50% will be purple. So if a heterozygous purple petunia, all right, so let's just do the cross. Heterozygous purple, it's only one allele, so we don't even have to do this. Let me start over. Heterozygous purple petunia plant is testcross. Remember, testcross is always to something that we know, so that would be the homozygous recessive trait or white flower. What proportion of the offspring would we expect to be purple? Well, if we did the standard Punnett square, we would see that we would expect that 1/2 would be white and 1/2 would be heterozygous, which is purple. But remember, only half of those are actually going to show up. So it will be 1/2, right? 50% we would expect to be purple if it was complete penetrance. But only half of these are going to show the trait because it's only 50% penetrance. So it would be 1/2 times 1/2, which is 1/4, which is 25%. The recessive disorder PKU has 60% penetrance. If 2 heterozygotes for PKU allele have a child, so heterozygotes, I'm just going to do the same thing. So 2 heterozygotes for PKU have a child. PKU is 60% penetrance, so we just do the Punnett square. Or you could just do this as a monohybrid cross, right? So you would know that the ratio is 1 to 2 to 1, which would be big P to big P to little p, little p. This would be the only way, so we would expect 1/4 if it was 100%, right? Penetrance, but it's only 60%. So we would times 1/4 by 0.6. So 1 divided by 4 is 0.25 times. We could just do this as 6/10, so that would give you 6/40, which would reduce to 3/20. 3 divided by 20 equals 0.15, which is 15%. Alright. Capybaras. So capybaras' fur color is determined by 3 alleles of a single gene, which is the following dominance relationship. So this. So these are red. This is red. This is black. And that one I assume is white. So we have the hierarchy here. So a red capybara, red, is crossed with a black capybara. So we know that the red ones can be big R, little r, B. That would still be red because this is superpowering over that. It could also be big R, or it could be big R, little w. Any of these outcomes would still make this thing red. So we don't know about this parent. It's a question mark. Cross with the black one. So the black ones, it can't be any of this. It has to be either homozygous for black, or it has to be that. Right? This is the black capybara. That's its genotype. We don't know. But this tells us something, right? That means that in order to be white, you have to have both white alleles, right? And to do that, then it has to be this, and it has to be this, because in order to get white, you have to have this offspring. So we can immediately get rid of that and get rid of this. And we know that it has to be this one, and we can get rid of this because it also has to be this one. So this is the red one. This is the black one. And if we do the cross for their offspring, so I'm going to move over here. So we do big R and little rw, standard Punnett square. I'm going to do it on this side, though, because I can't write on the other side. This is little rb, and this is little rw. We do the cross, and this one would be, big R, little rb. So what color would that one be? Red, right? This one is little rw, little rb. So what color would this? Black trumps over white. So this one would be black. This one would be big R, little rw. So what color would this one be? This would be red. So both of these are red. So 50% red. This one is 25% black. And this one would be the only way to get white, which would be 25% white, which explains this outcome. All right, next. All right. Harry plans to marry Sally, conceived by artificial insemination. Harry fathers, having served as an anonymous sperm donor, is concerned he might be Sally's father. Given the following information, deduce whether Harry and Sally can be related. Okay, so we know Harry's mother has to give this. This is Harry. You can just make sure that Harry's father's what he says he is. In order for Harry to be O here, that we know these two. If that's really his father, it has to be, these both have to be AO. Right? And so it's possible that they could pass on this to give S. They could pass on this to give N, and they could pass on this to give O. So it's likely that Sally and Harry really fathered Harry. Now, what's the probability that wait, we wanted to do Harry's mother. So she could be, I'm sorry, let me start over. Damn it. All right. So we have Harry's father and Harry's mother, and we want to make sure they're Harry's parent. So this could be AO, this could be BO. This could be NN. This could be little s, right? So now we just got to make sure. So this is cool. This NN is also cool. And then this OO is also cool. So Harry and Harry's mother and father are probably Harry's parents. We know Sally's mother, right? And Sally. So we want to know if these two could have this kid. And so we know that this could be AO. And so the question is, can two O people have an O kid? This would be OO, and the answer is yes. Can two people that have both M and N alleles have an MM kid? And the answer is yes. And then the question is that can two heterozygotes for S have a big S kid? And the answer is yes. So the answer to this is yeah. Harry's father could be the father of not only Harry but Sally, so they could be related to each other based on this information. So the answer is A. They could be half siblings. Okay. AT2 means that it has 2 repeats. AT5 means it has 5 repeats. Which lane of the gel shows the individual that's homozygous for AT5. Remember we talked about smaller pieces go further. So this would be AT2. Bigger pieces would not go as far. So this would be AT5. And because the gel can only separate based on size, these are the exact same size. It would look like one individual band, even though there's 2, one from each chromosome. So the answer is B. That's just showing you. This is AT5, AT5. This is AT5 and AT2. This is AT2, AT2. What's the sequence of this? Super easy. A. We just read from the bottom up. A, G, G, C, C. And remember, this is 5 prime. A, G, A, T. G, C, T, G, C. T, G, C, T, G, C. Oops. I wrote that twice. So that's the answer, and then this is 3 prime. All right. What are the 3 basic steps of conventional PCR? Remember, we have to heat up the DNA to separate the strands. And so that's called denature. And then we cool to bind the primers, and that's called annealing. And then the last one is we heat up to the TAC enzyme, the polymerase, optimal temperature, which is the temperature that it lived in those hot springs, which is 72, and we call that extension. So denature, the DNA comes apart, annealing of the primers bind. Extension is that the polymerase makes copies of the DNA. So the answer is B. Which of the following is not required for PCR? We need the enzyme. It can't copy without it, so we can't do it without the copy machine. We need the primers. Remember, that provides it with double-stranded. We talked about a normal regular train that needs 2 tracks can't ride on a single railed track. It would fall off. We need the parts, right? The A's and the G's and the C's and the T's. And we also need the template, the original DNA that we're going to copy, right? So this could be from your cat, or it could be from you, or maybe we're targeting the virus, that HIV that would be in your blood. But regardless, that is, we're going to need the DNA template, which is that. So we don't need RNA transcriptase. This is used to convert RNA into DNA. And honestly, we haven't even talked about that. So the answer is B. Which is more likely to lead to cancer? So remember, we talked about the cell cycle, right? And there are genes that are proto-oncogenes, and those give the go signal. And then we have tumor suppressors, and those give the stop signal because they suppress tumors, right? If we went through the cell cycle, you would get more and more tumors. So if our proto-oncogene lost its function, then it would no longer have the cell cycle go, right? So we wouldn't get more cells made, because every time we go through make a new cell, we have to go through the whole cell cycle. So that's not right. Gain of function of a tumor suppressor gene. So if a tumor suppressor gene gained its function back, it would suppress the tumor, and so you wouldn't get the cell cycle because it would tell it to stop. So that wouldn't do anything. If we gained function from a proto-oncogene, so if the proto-oncogene gained function, it would tell the cell to go, but and this could lead to cancer, right? Because it's going to cause it to go, and we don't want that. A loss of function of a tumor suppressor gene. If this was broken, then what would happen to the stoplight? It would say go, right? And so we would end up with green lights. So both C and D would lead to cancer cells. So the correct answer would be F. All right, sickle cell is caused by mutation of gene encoding beta-hemoglobin. Normal allele is this, mutant allele is that. Okay. So there was a slide actually on this. This is completely dominant over that, and so this is what we call haplo sufficient, right? Because in the heterozygote we get a normal phenotype. So normal blood count is dominant over anemia. That's correct. Great. This is a complete dominance. If it was incomplete dominance, then we would get partial anemia. Codominant, we would get partial anemia. So this is complete dominance. Answer is A. Partially sickle cell. Okay, so this could be codominant, right? Or incomplete dominance. Remember, the difference between the two is kind of shady, but if we're looking at one phenotypic outcome, which is partial sickle cell shape, then we would say that is incompletely dominant. Because if it was codominant, we would see the cells being two different things. Like in the blood group, we have A antigen on the surface and B antigen on the surface at the same time. So we would see both A and B. Because this is a blending of these two, right? Like we talked about with the snapdragons, where the red and the white equal pink. That is an incomplete dominant, not a codominant, and it's not dominant. So the answer is B. If you shrunk yourself down to the cell level where the proteins were made, then you can maybe argue codominance. But you better, if you put this, you better tell me that you're shrinking yourself down to the level of where the protein's being synthesized to call it codominant. All right. All right. So we're getting both, right? And so remember we talked about incomplete dominance is a blending. This is where we're getting two independent phenotypes. So in this case, we would call that codominant. So this answer would be C. All right, AT5. We know that's repeated 5 times. It's 0.3% of the population. AT7 is 0.1% of the population. What are the frequency of the heterozygote with AT5, AT7? So Hardy-Weinberg is this one would be P, this one would be Q, and we want to know the heterozygote, so that's 2 times P, which is 0.3, times Q, which is 0.1, so we just do the math. 2 times, oh shoot, I did that wrong. 2 times 0.3 times 0.1 equals 0.06, so it's this C. Which of the following mutation is likely to be neutral? Single base mutation of 5' end of an intron sequence. Deletion of 5 base pair part of an exon. Remember, introns get cut out, exons stay in. You delete part of an exon, that is going to cause a mutation. Probably, almost definitely. We add some bases and a repeat of a microsatellite. That's not going to do anything. Remember, microsatellites are generally found in the regions that aren't important. Non-synonymous mutations. Non-synonymous means that it causes the amino acids to change. So this would be bad. So this would cause a mutation. It wouldn't be neutral. This would I'm sorry, shoot. So we're going to exclude this because it can't be neutral. That would be bad. This one, we're going to exclude this one. That would be bad. The 5' end, so that's the intron exon junction. Remember, that's where we have that sequence that's super conserved. So we're going to exclude this too. And so the correct answer would be C. W spotted wings. Plasma does not affect fitness. Which of the following statements is true? Frequency of W allele in the population. W equals 0.1. So big W equals 0.9. WW is spot wings. It's recessive. So this normal. This would also be normal. So what assumptions can we make? This isn't telling us 90% of flies have no spots. What it's telling us is that 90% of all of these have at least one W, right? If we want to find that out, we have to do Hardy-Weinberg. So we know that W is 0.1 big W is 0.9. So we can calculate the percent that's going to be big W, little w, because that's 2 times PQ, which is 2 times 0.1 times 0.9. So 18% would be heterozygous, and then homozygous dominant would be 0.9 squared. 0.9 times 0.9 equals 0.81, which would be 81%, plus 18% would give us 99%. So that's wrong. That can't be right. We know that number is actually 99%. And if that number is 99%, then we know that this number is 1%, so we don't even care about the rest of this. That's the only actual answer that can be correct. All right. Normal blood, infected malaria. Yeah, okay. 9% of an African population is born with a severe form of sickle cell anemia. So we know that Q squared equals 0.09. So what percent is of the population is heterozygous? In other words, what percent of the population is 2 times P times Q? We don't have Q yet, but we can get it. Q is the square root of 0.09. Which is 0.3. And so if Q is 0.3, then P is 0.7. And now I just got to plug it in. 2 times 0.3 times 0.7 equals 0.42. That's what I get. So 42% of the population would be expected to be heterozygous or 2PQ. All right. VNTR, chromosome 8, repeat 719, TCA. We talked about it's ethnically diverse people, and different ethnicities have different frequencies of alleles in the population. You determine an individual's genotype is TCTA7, TCTA10. 7 repeats, 10 repeats at a locus, single locus. So this is 10% of the reference population. So let's just say that could be Caucasian or whatever. And then TC10 is found at 5% in the reference population. Well, I've already given you the percentages. So it's you want to calculate what the probability someone from that same ethnic group has that genotype. Then all you have to do is multiply it by 2 times P times Q, right? Because this is the probability that they're heterozygous for this trait. So what's the P value? 2 is, P is 0.1, Q is 0.5. So that gives you a value of 0.01, which is 1%. This one. Okay, same. It's basically the same question, but they're homozygous for this, and so we have this as 5%. So this would just be P squared, which is 0.5 squared, which is 0.0025, which is that value there. Okay, forensic scientists, blood cells match DNA profile, 5 different VNTR. So Hardy-Weinberg frequency of the DNA profiles for the 5 individual. So this is the key. It's telling you already the Hardy-Weinberg frequency, so you don't have to calculate it like we did in the last problem. It's already given to you. Someone already did that math. All right, so 5 loci, this one, this one, this one, this one, this one. How common or rare would this 5 locus profile be in the reference? Super simple. All we have to do is multiply this times this times this times this times this, because the table R has already given you the Hardy-Weinberg frequencies for those alleles. You don't have to calculate it out. So if you multiply 0.01 times 0.02 times 0.06 times 0.1 times 0.03, you should get this value here, which is C, which is 1 in 27.8 million, roughly. ANA 0.8, 0.2, assuming these are neutral, no additional mutations, what is the probability that A becomes eventually fixed in the population? Well, remember we talked about the probability of little a getting fixed is the exact same as its frequency in the current population, so that would be 20%. Unrelated givens, 4.4% nucleotides are different. So we can do the n equals pi divided by 4 times the mutation rate to calculate this. No, I don't want you to just restart now. This is an awful time. Great. Go away. Stop looking at me, Microsoft. All right, the mutation rate. We know it's... this value is 10 to the negative 7th, right? We're going to multiply it by 4, and then this is the heterozygosity. This is the effective population size. This is the heterozygosity, this value, which is 0.4, which is given. So all we do is divide 0.4 times 4 times 10 to the negative 7th. That's going to give us the effective population size. And wait. This is a different slide. It's giving you different numbers, but the answer is still the same, right? So we do 4 times 0.2. All right, so 0.4 divided by 0.000004 equals oh, that's 0.4%. I just made that 40%. That's not right. Let me change this. That's 0.004. All right, let me redo the math. So we have 0.004 divided by 0.000004 equals C, 10,000. Okay, my bad, sorry about that. That's just showing you how to calculate it. We talked about that. Which are not true of non-synonymous. So non-synonymous mutations, yeah, they can be neutral, rare cases. They're usually bad. Non-synonymous mutations are deleterious in a certain gene. The rate of base association non-synonymous traits revealing synonymous sites in this gene. So the question is, which of these are not true? This is not true. This is true. We talked about, and we have small populations, bad alleles get fixed. And what do they get fixed in by? Genetic drift. So the answer here would be B. And that just talks about it. DNA sequences continue to change due to neutral mutation and genetic drift. The rate of change is, remember we talked, it's equal to the rate of the mutation, which is mu. This is the molecular, this is just showing you the mutation rate is equal to the heterozygosity value. New non-synonymous mutations on side of heredity are more likely to be deleterious than those on, yes, of course. This doesn't change as much. This one does. This is the same slide that we did in class. So cytochrome C mutations are bad. That's why they don't happen very often. This doesn't seem to really care that much. So the correct answer for this would be A. Negative selection. What is the relative fitness of little a? So it's 1 minus S, right? And we have the S value as 0.3, so 1 minus 0.3 equals 0.7. So its fitness level would be D, 0.7. So remember, this one is 1 minus S. This is 1 minus HS, because we have to multiply it by the dominance, if it's codominant or complete dominant. Ancestral gene duplication. So these, all right, A2, and B1, those are paralogs, that's true. A2 and B2, they're in the same species. So they're still, these are paralogs, so that's not true because they're the same. They're different groups. Like we talked about hemoglobin A and hemoglobin B. All the hemoglobin Bs are in the same family. A1 and A2 are paralogs. A2, that's not true. These are orthologs. These are paralogs. This one is true. B1 and B2, that's not true. Those are orthologs. So the answer is A. And this just shows you what we talked about. Orthologs are the same group, like A and B, and then paralogs are different. So if we go back a slide, same, right? Those same ones are orthologs. So same orthologs, different paralogs, different paralogs. All right. Covariance. Oh boy. All right. So we're doing the covariance because we have number of hours of study and the average grade. So variance of the grades. So we'd need to do the means. 72 plus 84 plus 90 plus 94 divided by 4 gives us 85. That's the mean of all the grades. Where's my mouse? All right. I don't have time to do all this math, honestly. But I did it here. So here's the average, right? It's 85. And then we do the variance. Remember, it's the sum of the mean minus the actual grade squared divided by n minus 1. We get 169 plus 1 plus 25 plus 81, which is 276 divided by 3, which gives us 92, which is right there. So square root of 92, that's our variance, is 9.592. Covariance is the sum of the multiplied values of x and y. So 6 times 72 plus 9 times 84 plus 12 times 90 plus 15 times 94 gives us this value. Remember, we do, that's minus 1 divided by n, and the sum of the values of x times the sum of the values of y divided by n minus 1. So if the sum of x is 42, sum of y is 340. 42 times 340 is 14,280. So we plug it in, we get 3678 minus 1/4 times 14,280 divided by 4 minus 1, which is 36. So we can say that there is definitely a relationship, because that's a 36 is a positive value, and it's an actually pretty large value. We could calculate the regression value, but I'm guessing that's about a 0.9% regression. So there is a really strong correlation between the number of hours spent studying and the average grade in a typical genetics class. Okay, so we can do this one. We were given the environment, the additive, and the dominance effect. And we talked about this in class, but we did pumpkins, right? So we're doing frogs now. We want to breed frogs that have I guess we want to find out what the weight of the new groups of frogs if we pick parents that are 600 grams frogs and their normal average weight is 500 grams. So what's the genetic variance? So VG equals, remember we talked about VA plus VD plus VI plus V genetics in the environment. This is 0. This isn't given. So VG equals VA plus VD. So to calculate genetic variance, we just do VA plus VD, which gives us 1280. All right, let's erase all this. Okay, so the next question is, what's the phenotypic variance? Well, phenotypic variance equals the genetic plus the environmental. We already calculated the genetic is 1280, and we're given the environmental is 1340. So we just had to add the two together, and we get 2620. So that's the phenotypic variance. I'm going to erase that. It's down here at the bottom in the answers, by the way. And then, let's see, broad sense heritability. So big H squared equals VG divided by VP. We already have both these values, so we just have to plug it in, and we get 0.49. So that's broad sense heritability, right? Because broad sense is every genetic component. That's why it's called broad. The next one is narrow sense. Narrow sense is just narrow because we're only taking into account the additive variation in the population. And again, we have all the values that we need here. This is supposed to be a small h. I don't know what the deal is with that. All right, so that's a small h. And then we have VA over VP. We've already calculated it as 2620, so we get 870 divided by 2620, which is 0.33. And then if we want to find out the mean weight of the next generation, remember we have the narrow sense heritability value, which we already have, which is 0.33, equals the next generation, so what the frogs are going to weigh, minus 500, which is the original, divided by the new parents, right, which is 600 minus 500, which is 0. So that is 0.33 narrow sense heritability equals X, which we're trying to find out, minus 500 grams. I guess I should be using units. It's kind of bad form. Divided by 600 grams minus 500 grams, which is 100. So we get 0.33 equals, and then we can do X minus 500. So we just cross multiply that, and we get 33 equals X minus 500. Then we subtract. We add 500 to both sides, because that's a negative 500. Damn it. Wait, where was I? This is a negative 500, so we add 500 to both sides, so we get 500 plus 33 equals X, so X equals 533, which is this value here. Okay, perfect. All right, so that's it. That's the review. And I hope that this saves, because I will freak out if I spend an hour and a half not doing this. Right? Okay, so